Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, a very warm welcome to the TV learning programs. As usual, I'm your teacher Herbert, and I will be taking you through the biology and health science, senior 3. And we are still on the same unit, which is unit 8. This is supposed to be circulatory system in humans. And the lesson is lesson 4, which is concerning about blood clotting and even blood group transfusion. So I think last time we talked about composition of blood or components of blood. We said there are a number of components of blood. We talked about blood plasma, which makes almost like 90% of the body, of the composition of the blood. And we talked about the platelets. We talked about red blood cells, which is RBC. We talked about white blood cells, WBC. And we also talked about the platelets. And the platelets is going to be involved in that lesson of blood clotting. And we mentioned one of the functions of platelets, it plays a function in blood clotting, or as far as blood clotting is concerned. So let's talk about white blood cells. They are responsible for protecting the body against pathogens, that is disease-causing agents. We said red blood cells, or RBCs, are responsible for transport of oxygen and even nutrients to the different parts of the body or to the body cells. So we talked about platelets, which is also very important, and as far as blood clotting is concerned. So let's start our lesson, but before we start our lesson, I would welcome everyone to get a book, a notebook, and you start to note the important concepts or words which are involved in that lesson. Welcome to the lesson now. So the learning objective of the lesson, we have to explain how does the process of clotting of blood take place? How does the blood clot when a person gets damaged or when a person gets injured? We also have to describe blood group transfusion. So that is what we're going to be discussing today, and I welcome you all in the lesson. So before now we are going to start, what is blood clotting? I think in the previous lessons we talked about blood clotting, and we talked about sometimes when the blood clots or when the blood clamps in the blood vessels, it can just be very fatal. It can just be very dangerous because it can cause what you call coronary heart diseases. But now we're going to discuss how the blood clots when a person gets injured or when a person gets damaged. So blood clotting is a seal that forms to close the blood vessels that are cut or damaged. That seal that is formed when a person gets damaged, when a person gets injured, that seal that is formed on our skin, that one is referred as blood clotting. And that one stops further bleeding at the wound and therefore prevents excessive loss of blood. When that clot is formed on the skin, what happens? This stops further bleeding. It stops for further bleeding. You no longer bleed. And that seal, that further bleeding, that would and therefore prevents excessive blood loss, because someone can bleed to the extent of death. So if there's no blood clotting. So blood clotting is very important in order to prevent excessive loss of blood. I think you are getting what I'm talking about. It also prevents the entry of harmful bacteria into our body through the damaged tissues. So blood clotting is also very important in preventing the entry of bacteria or any microorganism into our body through the damaged tissues. Because once we get damaged, eventually some of the microorganisms, some of the bacteria can enter through the damage. So blood clotting is also very essential to prevent the entry of bacteria into the damaged tissues. Let's have a chart that tries to show how does the blood clots takes place. I think we can just go through the diagram and we try to see and observe clearly. We have someone that is, this is maybe the skin, a damaged tissue, a damaged blood vessel, a tissue. You have a damage on a blood vessel, or you have a damage on a tissue. What happens? Automatically, once you have a damage on a tissue or on a vessel, on a blood vessel, what happens? That one stimulates the platelets gathered at that site of injury. The platelets will have to be gathered on exactly where the damage is, And once the platelets are gathered at the site of injury, what happens? The second step is to stimulate what you call a thromboplastin. We have what you call a thromboplastin. Once the thromboplastin is going to be produced, it is going to be, I said, once the platelets gathered at the site of injury, and that one stimulates an enzyme which is called there is what you call thrombokinase. That thrombokinase will stimulate, will activate the production of what you call thromboplastin. Once the thromboplastin is released, we are on step 2, on third step of blood clotting. Automatically, the thromboplastin will stimulate the release of the calcium ion. And that's why we say some of the mineral ions are very important in blood clotting. Take an example, the calcium ion. Calcium ion is very important in blood clotting because it stimulates what you call prothrombin. A prothrombin is a soluble protein, and even what you call vitamin K, which plays a role in blood clotting. After calcium is being released, which activates some enzyme to stimulate the production of prothrombin and even vitamin K. Now, the second step is what we call production, what we call thrombin. After production of the thrombin, it produces what we call a soluble vitamin. That soluble vitamin is called what you call fibrinogen. And once the fibrinogen is produced, it will just be converted into insoluble protein. That soluble protein is referred to what you call fibrin. And now fibrin is going to be released, which is a clot, and forms what you call, I can make it to be, this is supposed to be like a seal or a network of what you call fibrin, which clots the blood and stops the blood to be released again or to be continuing bleeding. And now eventually the bleeding stops. Once the bleeding does not stop, it means that the person is lacking what we call platelets. Lack of platelets result in what we call bleeding without stopping. I think we have seen the chart of what we call a blood clotting process. I said once the damaged tissue or a blood vessel is damaged or a tissue, what happens? It triggers the platelets to be gathered at the site of the injury. And after they are gathered at the site of the injury, what will happen? An enzyme which is called thrombokinase will stimulate to release what we call a thromboplastin. Once thromboplastin is released, it also triggers the release of the calcium ions, which activates some enzymes that are involved in what we call production of what we call prothrombin and even vitamin K. So the production of prothrombin and even vitamin K is as a result of what we call the release of calcium ion. So meaning that the absence of calcium ions, prothrombin and even vitamin K will not be released. And once the prothrombin and the vitamin K are released, now it converts into what you call a thrombin. And now from thrombin to what you call a fibrinogen. And I said fibrinogen is insoluble protein. And now the insoluble protein is going to be converted into a soluble protein which is called fibrin, clot, which is a seal that prevents the blood from bleeding. Let's go through the process as I was talking about. When blood vessels are damaged, or for example, you've got a cut on the skin. Maybe you have stepped on a nail, and the nail pinches you, and automatically the blood will start bleeding. The damaged tissues and platelets are released. A chemical code that are normally released by what you call a prothrombin. So prothrombin or thrombokinase are the one that triggers the release of what you call platelets. These substances convert blood protein called prothrombin to an enzyme which is called thrombin. That enzyme, which was released because of production of what you call calcium ions, which are going to be converted blood protein called prothrombin to an enzyme which is called thrombin. A thrombin changes a soluble protein, a soluble blood protein clearly, which is called fibrinogen, into insoluble fibrin and a mesh of fibers which traps red blood cells. So the red blood cells are no longer to be passing through because there's a mesh, what you call fibrin, which prevents the blood from leakaging or so to continue bleeding. So that is the process of bleeding. So this generates a clot, which shrinks as it forms, and hence pulling the edges of the wound. The edges of the wound will just be pulled together and assist in sealing it. And it dries up to form a scab, which protects the wound and giving a tissue beneath time to heal. And vitamin K is needed for prothrombin to be formed in the liver. So absence of vitamin K, the prothrombin will not be formed in the liver. So for the process of prothrombin to be formed in the liver, there must be a release of vitamin K. So let's go to another thing we've been talking about, blood group and blood transfusion. I know all of you know there are almost 4 types of blood groups. Types of blood groups. Types of blood groups. I know all of you, either in your family, some of you know the types of blood groups that you possess, and also the type of blood groups of your relatives, your siblings. You know there are types of blood groups. We have almost 4 types of blood groups. We have blood group A, we have A, we have B, we have AB, and we also have O. We also have another type of blood group, but today we're not going to discuss it, which is called a rhesus factor. It's also another type of blood group. So we're going to go through everybody. Everybody's blood is classified into blood groups. Our blood is classified into blood groups. Student, I know you know what is called blood, and everybody's blood is classified into blood groups, or what you call blood type. So the red blood cells of human have a special type of protein which is called antigens. In the blood we have antigens, and there are many types of such antigens, including anti-A, we have anti-B, we also have an antigen that the antigens are the one that determines the blood group. Antigens, these are special type of proteins that are found in the blood. We talk about antigens. And antigens must be compatible with antibodies. We have anti-A, we have B, we also have A and B, okay? So these antigens, they are the one that determines the type of blood group. Are we together, student? I will come to this lesson. Is very interesting and very, very, very essential to know. Everybody must be knowing you have a type of blood group. There are many types of such antigens, as I mentioned it, including anti-A, anti-B. Antigens determines the blood type or the blood group of a person. So what determines the blood group of a person? What determines the blood group of a person is the antigens. So antigens determine the blood group type or the blood group of a person. A person with only antigen A on their blood cells are said to be blood group A. If I have antigen A, another person must be having antigen B. Eventually, a person with antigen A must be having blood group A. A person with antigen B must be having blood group B. All right? Then a person with antigen A and B, antigen A and B, when he has both antigen A and B, the person must be a blood group AB. Then when it comes to blood group O, blood group O has no antigens. So that's what I was talking to you, that our blood group, most of the type of a person with only antigens A on their red blood cells are said to be blood group A. A person with only antigen B on their red blood cells are said to be blood group B. So people with antigen B are only belongs to blood group B. So if you have an antigen A in your red blood cells, you are a blood group A. When you have an antigen B in your blood group, you belong to a blood group B. So I also mentioned that if you have antigen A and B, the person must be having blood group A and B. Sometimes both antigens A and B are found on both normally found in both red blood cells. When you find that you have both antigens in your red blood cells, you are belonging to a blood group AB. In other people, the blood that has no antigens on their red blood cells, such a people are said to be blood group O. So if you don't have antigens, if there are no antigens or there are non-antigens in your blood group, that is I'm mentioning about A and B, it means you are belonging to a blood group O. So I precise that I can also conclude by saying that antigens are the one that determines the blood group. Let's have the table, antigens and blood group. We have the table, we have antigen present on red blood cells, we have blood group. A person with antigen A must be having a blood group A. A person with blood group B must be antigen B, a blood group antigen B present in the red blood cells. If you find in your red blood cells you have antigen B, it means that you have a blood group B. When you have antigen A and B in your red blood cells, it means that you are belonging to a blood group AB. When you don't have any antigen in your blood group, it means that you have a blood group O. In addition to the antigens, to the red blood cells, blood plasma contains other types of proteins. Not only that antigens are only types of protein found in the blood, you also have other types of proteins that are found in the blood. Those types of proteins are referred to as antibodies. When you have antibody, let me try to mention it. When you have blood group A, let me say this is a blood group, and here we have antigens, and here we have antigens and we have antibodies. So you have to go through this and you understand it very well. A blood group A, as mentioned, a person with blood group A has an antigen A, but has an antibody B. A blood with blood group B has got antigen B, but has antibody, which is against antigens, because antibodies and antigens are compatible. So we have antibody A. Then a blood group AB, as I mentioned it has antigen A and B. You are following me very well. But has antibodies, there are no antibodies. None. There are no antibodies. That's why we consider blood group A to be a universal recipient, because there's no antibodies. Then a blood group O, it has no antigen, there are none. Okay? There are no antibody, there are no antigen, it has no antigen, but has antibody A and B. Antibody A and B. So that is a simple understanding of what I'm talking about. Let's go through the chart. In addition to the antigens, the red blood cells and the blood cells, blood plasma contains other types of proteins called antibodies. These are complementary to the antigens. The antibodies must be complementary to the antigens, and they're supposed to be anti-A, and must be complementary to antibody B. Then antibodies are named A and B. So antibodies, they are named A and B, and they are rated respectively. Antigens and antibodies that correspond to each other are never found together in the same individuals. So antigens and antibodies that are the same, they are correspond, let me say, ant-A and ant-B. When you find the antigen A having antibody A, they must be not corresponding to each other, and they are never found together in the same individual, which can result in what you call agglutination. So let's say a person with antigen A will have antibody B in the blood plasma. A person with antigen B will have antibody A in the blood plasma. As I mentioned it, if both antigens are present, as in case of blood group A, then there's no antibodies. There are no antibodies. As I mentioned it, there are no antibodies. Then, as I mentioned, if none of the antigens are present in both antibodies are present, this is the case of blood group O. It means that if none of the antigens is present, then both antibodies, the antibodies I'm talking about A and B, are present. This, in that case, when you have this A and B, this means that the blood group is blood group O. Let's have a chart that shows how these antigens and antibodies are corresponding. We have blood group, we have A, B, AB, and O. I think everybody we are together with me. A person with antigen A in their red blood cells, he must be having antigen A, and he must be having antibody B, and in that we have, for the case that's supposed to be, let's have on that side. This is blood group A, must be having antigen A and an antibody B. A blood group B must be having antigen B and antibody A. A blood group AB must be having antigen A and B and now no antibodies. A blood group O has no antigens but has antibodies of A and B. As I mentioned it here, this is a summary of it. You try to go through it until it will be well understood. Well understood. You have to go through that, and you're able to understand. This is the chart that summarizes each and everything I've been talking about. A blood group A must be a blood group A person must be having antigen A and an antibody B. A person with blood group B must be having antigen B and an antibody A. Then a blood group AB must be having antigen A and B, but no antibodies. A blood group O must be having no antigens, but an antibody A and B. So that is what we've been talking about. Let's talk about blood transfusion. If the person who's voluntarily goes to the hospital or any health center to give blood, you are able to donate the blood. Blood is taken from the donor through a vein in arms and into the bag, what you call blood bank. After the blood is picked from a person, it is going to be taken to the blood bank, able to be tested if it is safe. This is kept in what you call blood bank. The word is here, under a suitable condition to be given a patient who needs it. We have donated blood is introduced into arms, and even as you're doing it, we have, when the blood is transferred from one person to another person, that one is referred to as the blood transfusion. The process of putting donated blood into a recipient is known as what you call blood transfusion. The process of putting the donated blood into the recipient is known as blood transfusion. So let's go to what is required when you are doing what you call blood transfusion. There are a number of processes. Blood transfusion may be necessity by situation such as, why do we need, when it comes to a need of blood transfusion? When a person loses too much blood, maybe due to an accident or due to an injury, may result into maybe from a motorcycle, a motor accident, falling down, fighting, or and other cases can lead into loss of blood. And in that case, a patient may need blood. When a person becomes anemic, that is when the red blood cells coil and they are no longer having too much blood. When a woman loses too much blood during childbirth, also needs blood. Sometimes, some of the situations that may cause one of the need of blood is what is preventable. You can't prevent it. Young children may expect mother catch and malaria. When you're suffering from malaria, red blood cells will also burst. And once they burst, what happens? You also be eventually in need of blood. So successful blood transfusion is one of which a recipient, a body will accept or donate blood without agglutination. When you give a blood group A must get blood from A and also gets a blood from O. A blood group B will get a blood from B, from himself, and also gets blood from OB. Blood group A will receive blood from all blood groups. All blood groups, and then also donates blood from A and B. Blood group O will donate blood to all these people, and also donates blood to himself. So that's what you're talking about. That is what you're saying. A blood group O, A, that is, yes, he can donate to this one, yes, O can donate to A, to B, and to AB. A to this one and this one in that way. That is A, yes, no. Blood group A cannot donate to O, but can donate to A, cannot donate to B, N stands for no, and can also donate this one. That is the table that summarizes what I was talking about. So once a person receives a blood, donates a blood to all blood groups, it is referred to as a universal donor. Once a person receives blood from all blood groups, it is referred to as a universal recipient. And if the blood is not compatible, it can result into what you call agglutination. That is a table, and that is home assignment. You have to go through that home assignment. And I think everybody is able to do those numbers. Number one is if one of your parents' blood group is type A and another is type B, which of the following blood types are you likely to be? We have A, B, B, C, C, and D, and AB. So you have to go through that exercise. There are almost 4 numbers. So do the exercise, and I hope to go through with you in the next lesson. Enjoy the rest of the day. See you next time.